everyone. Welcome back to SHK Creations YouTube channel. So in this video, we are learning about what is cell, the structure of cell and the functions of each organelles in cell, particularly animal cell and plant cell and many more. So let's begin. So what is a cell? A cell is a structural and functional unit of a living organism. This cell is a membrane-bound basic unit of life which is responsible for all the life processes. It may be digestion, excretion, reproduction, absorption, and for life. So who discovered the cell? Robert Hooke is a scientist discovered the cells in 1665. He was a British scientist and he discovered cells and he observed the cells on thin slice of cork under a microscope. What are the basic organs of cell? The basic organs of a cell, also known as organelles. These cell organelles perform specific functions both in case of plant cell and animal cells. The cell organelles like mitochondria, ribosomes, endoplasmic reticulum, nucleus. So we are studying in detail in future slides. Coming to the primary functions of a cell. A cell provides structure and support for an organism. Fine. They maintain the shape by providing mechanical support. And growth. The cells undergo mitotic divisions, meiotic divisions, like they help in reproduction. And they transport the nutrients. They regulate the movement of nutrients, waste products, and other substances across the cell membrane. And the cells involved in energy production. So ATP, adenosine triphosphate, it is the energy currency of a cell. So because of various metabolic reactions that takes place in whether it may be in cellular respiration, the production of ATP takes place, and like that the cells involved in energy production, which are very necessary for a life. Response to stimuli. See, if we touch hot water, we take our hand back. And in case of plants like touch me not plant, they are giving response to the stimuli. So the cells, certain cells are responsible for this, that is cellular communication. So the cells can detect and react to the changes in the environment through signaling pathways. So excretion, yes, our skin is also an organ of excretion through sebaceous glands. They will those glands involved in secretion of the waste. And nutrition, taking in nutrients from the environment that can be used for energy production. And majorly differentiation. See, in future, in coming slides, we can learn that stem cells can differentiate into any kind of cell. So these are like very basic functions of cells. Coming to the structure of cell. So plant cell and animal cell. So there are some certain differences. So mainly some organelles which are present in animal cells, they will be absent in plant cell. Fine. So the major difference is this is a vacuole. This vacuole is only present in case of plant cell, whereas this vacuole is absent in animal cell. And chloroplast. The chloroplast, as we know, it is a pigment, chlorophyll. These chloroplasts are organelles which involve in photosynthesis. Uh, these plants are autotrophs, so they can produce their own food by the process of photosynthesis. So this chloroplasts are organelles which are responsible for conducting photosynthesis in case of plants, but these chloroplast organelles are absent in case of animal cells. So mainly going in detail with the cell organelles. So let's differentiate between plant cell and animal cells. Which are all the organelles which are absent in case of plant cell and what may be the differences. So plant cell usually are large than animal cell. These animal cells are comparatively smaller than plant cell. So mainly the cell wall is present in case of plant cell in addition to plasma membrane, and it also consisting of middle lamella and primary and secondary walls. See, that's why we can see the hard layers of plant and trees, so because of the presence of cell wall, but these cell walls are absent in case of animal cells. So plasmodesmata is present in case of plant cell, while the plasmodesmata is absent in animal cell. These plasmodesmata are just like channels that connect plant cells and allow the exchange of materials between the cells. So as I said previously, chloroplast is absent in case of animal cell, and vacuoles are large and permanent in case of plant cell, whereas vacuole is small and temporary. But like storage vesicles are present in case of animal cells, but not particularly a vacuole, a large vacuole which is present in case of plant cell, and tonoplast present around the vacuole. Tonoplast is a membrane that surrounds the central or large vacuole in case of plants, but this tonoplast is absent in case of animal cell. And centrioles are absent in case of motile cells of lower plants, but the centrioles are present in animal cells. The centrioles are very important organelle which help in cell division and duplication, and also you might have studied the arrangement of spindle fibers also takes place during meiosis. The centrioles takes a role, and nucleus is present along the periphery of the cell in case of plant cell, but the nucleus is located at the center of the cell. In case of plant cell, the lysosomes are rare, but animal cell, the lysosomes are present. The lysosomes are called suicidal bags. In plant cell, the storage material is starch grains, whereas in animal cells, the storage materials is glycogen granules. These are just differences between plant cell and animal cells. Coming to cell membrane. So cell membrane gives a structure and framework for a cell. This cell membrane is also called plasma membrane, and it acts like a wall and serves a clear boundary between the cell's internal and external environment. And the main feature of this cell membrane is it is selectively permeable. It doesn't allow every substances into the cell. It only allow based on the availability of a cell. So it is said to be selectively permeable. And the cell membrane are made up of fat-based molecules called phospholipids and proteins. They provide structural support. They form channels for passage of materials, and they act as marker and functions as a communicator by sending and receiving signals from neighboring cells and environment. Fine. And the very famous widely accepted model of plasma membrane or cell membrane is the fluid mosaic model, which is proposed by Singer and Nicholson in 1972. So what is a fluid mosaic model? And let's see in this figure. So as fluid mosaic model says, the structure of plasma membrane is made up of phospholipids, proteins and other molecules. So coming to phospholipid bilayer, the foundation of the cell membrane consisting of two layers, as we can see in this picture, the phospholipids, this plasma membrane made up of heads and tails. This is phospholipid bilayer. So the foundation of the cell membrane, mainly this, consisting of two layers of phospholipid molecules with hydrophilic heads facing outwards towards the aqueous environment, and their hydrophobic tails. Phobia. We have phobia for something. Definitely we all be having like that. So we will face opposite. So this hydrophobic tails facing inwards, so creating a barrier to most water soluble substances. And then there are certain different types of proteins which are embedded in this phospholipid bilayer layer, and this protein perform diverse functions like transport channels, receptors for signaling molecules, and enzymes. See, as you can see, some peripheral proteins, globular proteins, integral proteins. Fine. So the integral proteins, they span the entire membrane, acting as channel or transporters. As you can see, this is an integral protein. They just span the phospholipid bilayer. And peripheral proteins attached to the surface of the membrane, they are often involved in cell signaling. And cholesterol. This cholesterol molecule present within the phospholipid bilayer, they contribute mainly to the membrane fluidity and stability. Glycolipids and glycoproteins. So carbohydrates groups attached to the lipids, they are glycolipids, and carbohydrate groups attached to the proteins, they are glycoproteins. So this we can see on the outer surface of the membrane. So these are glycoprotein and glycolipids. They are important for cell recognition and cell adhesion. Fine. Coming to the functions of cell membrane. As I previously said, cell membrane is selectively permeable. It is the ability of a plasma membrane to allow certain substances to pass through while blocking others to maintain the internal cell environment. And this plasma membrane facilitate the movement of molecules by certain mechanism. So one is active transport and passive transport. As you can see in this image, active transport required the ATP energy molecule to transport the molecule across the cell membrane, while the passive transport, it is based on the concentration gradient. One is diffusion and another is osmosis. So diffusion is the transport of molecule from higher concentration gradient to lower concentration gradient, while osmosis is a movement of water molecule across a semi-permeable membrane. So the receptors which are present on plasma membrane, they bind to signaling molecules. It might be hormones, neurotransmitters, then in turn which triggers the cellular responses and cell to cell adhesion. Those specialized membrane proteins, they enable cells to connect and interact with each other. So this plasma membrane will be having some adhesion proteins. So when ligand binds to the adhesion proteins, so this cell adhesion takes place. And also like plasma membrane is a boundary, it is like a boundary, so gives structural support to maintain the cell's shape. Nucleus is the control center of the cell. This nucleus consisting of dense material called nucleolus. This is nucleolus, and a thread-like structure, they are called chromosomes. Here the condensed form of chromosome, this can be labeled as chromatin, and it contains genetic information that is necessary for cell growth and reproduction. So that is DNA, deoxyribonucleic acid. This nucleus decides the activities of a cell, which carries the hereditary information. As it decides the activities of cell, it is called as a mastermind of a cell. This is a diagram of nucleus. And cytoplasm. Cytoplasm is a gel-like fluid inside the cell. It contains all the organelles, and it is like giving a space for organelles to fit in. It provides a platform upon which other organelles can operate within the cell. So all of the functions for cell expansion, growth and replication are carried out in the cytoplasm of the cell. Cytoplasmic organelles. So these little organelles are suspended in the cytoplasm of a cell. So each organelle have a very definite and particular function to perform. So like mitochondria, ribosome, Golgi, lysosome. So let's discuss the functions of each organelle. Nucleus is surrounded by nuclear envelope, and it is a double membrane. This nuclear membrane is perforated by nuclear pores. The nuclear envelope is continuous with endoplasmic reticulum. So this endoplasmic reticulum, again we can classify it as rough endoplasmic reticulum and smooth endoplasmic reticulum. So based on the presence of ribosomes, the main function of nucleus is like They houses a chromosomes made up of chromatin, which is a genetic material, and proteins. So they contain nucleoli where ribosomal subunits are made. So these pores regulate entry and exit of material. So these are the functions of nucleus, and mainly ribosome is the protein factory, which is responsible for the protein synthesis. Fine. So endoplasmic reticulum, it is like continuation of nuclear envelope. So based on the presence or absence of ribosomes, the smooth and rough endoplasmic reticulum made. So in case of rough endoplasmic reticulum, the ribosomes are present. Coming to the function, the smooth endoplasmic reticulum, synthesis of lipids, metabolism of carbohydrates, calcium storage, detoxification of drugs and poisons. So the function of rough endoplasmic reticulum, this aids in synthesis of secretory and other proteins from bound ribosomes, adds carbohydrates to glycoproteins, produces new membrane. These are the functions of endoplasmic reticulum. So Golgi apparatus. So this Golgi apparatus like stacks of flattened membranous sacs. See, these having cis and trans phases. The functions of Golgi apparatus like packaging of proteins like chemicals. So modification of proteins, carbohydrates on proteins and phospholipids, synthesis of many polysaccharides, sorting of Golgi products which are then released to vesicles. So these are the vesicles. So whatever the production takes place, they are released and sent in the form of vesicles. And coming to lysosome, lysosomes are called as suicidal bags. So they are membranous sac of hydrolytic enzymes in case of animal cells. Coming to the functions of lysosome, so they are suicidal bags because they degrade the damaged organelles, and they remove the ingested substances like breakdown. So breakdown of ingested substances, cell macromolecules and damaged organelles for recycling. So vacuoles, so large membrane bound vesicle in case of plants. So mainly they are meant for storage, waste disposal, water balance, cell growth and protection in case of plants. So this mitochondria is bounded by a double membrane, and inner membrane has a infoldings called cristae, as you can see in this picture. So these foldings are called cristae, and it is a place where cellular respiration takes place. And chloroplast, typically it is also two membrane structure and responsible for photosynthesis. Peroxisome, it is a specialized metabolic compartment bound by a single membrane. The function of peroxisome is they contains an enzyme that transfer hydrogen to water by producing hydrogen peroxide as a byproduct, which is converted to water by other enzymes in peroxisomes. Plastids are specialized organelles which found in plant cell. They are primarily responsible for the photosynthesis, pigment production, and storage of various food substances like starch, proteins, and lipids. So, based on the content and function, based on the storage of a substance and a function, the plastids are classified into many types. So, etioplast is a precursor of chlorophyll. It is a crystalline prolamellar body. And chromoplast, pigment synthesis and storage. Chloroplast is mainly responsible for photosynthesis, and leucoplast, synthesis of fatty acids and amino acids. Amyloplast is, as we can say, salivary amylase, amylo, so storage of starch and sugars. And elaioplast, storage of lipids, oils. Proteinoplast is site of enzyme, that is protein activity. And this is a chart that can sum up the functions of cell organelles. Nucleus, the cell's control center and contains DNA, hereditary information. Cell membrane, flexible gate and control what goes in and out of the cell. Mitochondrion, it is a cell powerhouse, provide energy to the cell. Golgi complex, package of proteins so they can be taken out of the cell in the form of vesicles. So endoplasmic reticulum transport proteins within the cell. Lysosome get rid of waste including worn out cell parts. Vacuole store the substances for later use. Cytoplasm, the fluid that everything in the cell is floating in. Ribosome, where proteins are made, that's why they are called protein factory. Cell wall, stiff materials that surround the entire cell and provide support and protection. Chloroplast, it's a green part where plants do photosynthesis, use sunlight to make food. So this is like a nickname for mito cell organelles. So mitochondria, powerhouse of cell, thylakoids, baggy trousers, Golgi apparatus, director of macromolecular traffic, post office or warehouse, traffic police of the cell, and lysosome, suicidal bag, disposal unit, atom bombs, demolition squad, scavenger of a cell. These are like nicknames. And ribosome, pallid granules, and protein factories of cell. Nucleus is control center or brain of cell. Nuclear plasm is nuclear sap or karyolymph, nucleosol or nuclear hyaloplasm. And is based on the presence of membrane around the nucleus or not. So based on this, the cells are classified into prokaryotic and eukaryotic cell. These prokaryotic cells we can see in case of bacteria. So mainly the nucleus is surrounded by a nuclear membrane in case of eukaryotic cell, but in case of prokaryotic cell it is not. So the main differences is no membrane bound nucleus in prokaryotes. So membrane bound nucleus is present in eukaryotes. So cell wall is made up of peptidoglycan. So the thickness of wall depends on whether the cell is gram positive or gram negative. So we can say it has a gram positive bacteria or gram negative bacteria. And based on the presence of peptidoglycan layer. And the cell walls, if present, it is made up of cellulose in case of plants and chitin. It is a cell wall component in case of fungi. No membrane bound organelles are present in case of prokaryotes, and membrane bound organelles, that is compartmentalization, we can see in case of eukaryotes. And in prokaryotes, so organisms have pili and fimbriae for adhesion and flagella for propulsion. So in eukaryotes also they have cilia or flagella for movement, and mucilaginous capsule which is present in case of prokaryotes. Here no mucilaginous capsule present in any of the cell. So numerous internal structure, as we can see, double membrane structures, organelles are present like Golgi, and they produce secretory vesicle, the endoplasmic reticulums are present. The cell size ranges from 0.5 micrometer to 100 micrometer, and the cell range of in case of prokaryotes, but eukaryotes the cell size ranges from 10 to 150 micrometer. These are the basic differences between prokaryotic and eukaryotic cell. So stem cells are cells with the potential to develop into many different type of cells in the body. So they serves as a repair system for the body. And the main two types of stem cells are embryonic stem cells and adult stem cells. See, they have the ability to make more cells like themselves, like self renewal. They can become other cells that do different things. That is differentiation. They can become any cell. So stem cells are found in almost all tissues of the body. These stem cells are different from other cells in three ways. One thing is they can divide and renew themselves over a long time, and they're unspecialized. so that they cannot do specific functions in the body. They have potential to become specialized cells, like they can become muscle cells, blood cells, and brain cells. Um, fine, as you can see in this picture, they can become any cells: liver cells, nerve cells, cardiac cells, blood cells. So this is known as differentiation. There will be some certain processes how they differentiate. So that you can study in developmental biology. And coming to applications of stem cells, so mainly regenerative medicine. So the stem cells can be used to regenerate uh, damaged organs and tissues, and they can use to treat the root cause of the problem, not just treating the symptom. Fine. Stem cells can be used to repair damaged cells and speed up healing. That is regenerative medicine. And organ transplantation. The stem cells can be manipulated to become specific cell types, such as heart cell, muscle cells, blood cells, nerve cells. They are specialized cells. And then the, the cells can be implanted into a person. A stem cells are bone marrow transplant. They can replaces the damaged blood cells within the healthy ones. It can be used to treat conditions affecting the blood cells such as leukemia, lymphoma, and in future it might be possible by using the stem cells to get a permanent cure about other diseases like Parkinson's disease, Alzheimer's disease, and many more. So the research and studies are going on that way. Drug testing. So stem cells can be used to test new drugs for safety and effectiveness. So they have to test, because before trying on human use, so they need to conduct some drug test so they can use the stem cells. Research. So stem cells can be used to research how diseases occur, or why certain cells develop into cancer cells. And stem cells can be used to research the cause of genetic defects in cell. And bioprinting. 3D bioprinting technology can be used to manufacture organ system derived from stem cells. And stem cell engineering, it is the use of techniques to manipulate and control the behavior of stem cells. So these are the important applications of stem cells. So this is all about the simple and beautiful concept in biology, that is the cell and its functions. I hope you all like this video. So if you like this video, please do like, share and subscribe and keep supporting. Thank you.